So our next speaker, and this is going to test my technical skills again, is speaking remotely. So the next speaker is Rachel Colquhoun. I hope I pronounced your surname correctly. Rachel, are you there?
I'm here, yes. Let me just share my screen.
And we can see and hear you, which is amazing. So if you could share your screen.
There we are.
Fantastic. Take it away.
Great, thank you very much. So firstly, thank you for inviting me to speak today. My name is Rachel Colquhoun. Some of you will know me as one of the people behind Bioinformatics and Datapipe. Way back in January 2020, we came into the office one morning to find that Andrew had taped this to Anya's monitor. We didn't know it yet, but this was the start of our work within the group on the pandemic, and it wasn't long before the whole group was working on different tools. The plan for this talk is to first give an overview of the tools that have been developed by various people across the consortium, bioinformatics tools that is, before going into a little more detail on some of the tools and methods developed within this group here in Edinburgh that have enabled us to scale the work we do to the huge volumes of data and gain some actionable insights. There has been a lot of bioinformatics done within the COG consortium because it really has been fundamental in every step along the process. I'm going to give a whistle-stop tour here, highlighting some of the key names I know about associated with each step, but in many cases, there's actually a team of people who contributed. Starting at the very beginning of the process, there's been work on the field bioinformatics software and the Arctic protocol for generating consensus sequences from nanopore sequence data, which has included Will Rowett and Nick Lohman's group. This has been built into a Nextflow pipeline by Matt Bull and others in Tom Connor's group over in Cardiff. There's the setup and management of the Climb IT infrastructure by Rad, Matt, and more recently Alex. And there's been the challenge for managing metadata uploads, which Anthony Underwood has masterminded. For sequences and metadata on Climb, Sam Nichols, Majora, and the ELAN pipeline have managed combining them into a single unified dataset, with additional pipelines automating uploads to GISAID and ENA. And more recently, Sam Wilkinson has joined to help keep these running. The ELAN pipeline feeds into the Datapipe and Bioinformatics pipelines, which are written and managed by myself and Ben Jackson. This performs further QC, alignment, variant calling, and lineage assignment, cleans up the geography metadata using scripts by Verity, as well as constructing a phylogenetic tree. And this central analysis then feeds into tools like CIVIT by Anya and Verity, which generates individual outbreak reports, MicroReact by David Arneson and Anthony and team, which allows the tree and some of the geographic data to be explored. COG-ME by Derek Wright and others over at CVR in Glasgow, which allows mutation profiles to be explored. And there have been software like Figtree 2 and Taxonium, which have been written by Emily Scher and Theo Sanderson, which enable the massive trees to be visualized. Datapipe outputs have also fed into larger individual analyses into questions like the origin and expansion of the first wave and the alpha and delta variants. This has required bioinformatics improvements to software Beast to enable it to scale nearly 2 orders of magnitude, greater than the largest previous analysis. Not to mention some of the better or less known standalone tools which have been written, including Pangolin for lineage assignment, Scorpio for constellation classification, Polecat to identify and flag phylogenetic clusters which could be concerning, and Grinch, which allows exploration of the spread of Pango lineages. So I think you'll all agree that's quite a lot, and I'm sure there's a whole lot missing from that slide, which could be on it. So I'm now going to focus the rest of my talk on some of the work we've done here in Edinburgh, touching on some of the ideas in Datapipe, Bioinformatics, CIVIT, and Thorny Beast. When COG-UK was set up in March 2020, the aim was to use the sequence data to understand viral transmission and evolution and inform public health responses in real time, both at the national scale via weekly reports to SAGE and at the local scale, at the scale of local NHS sites. We hope to be able to answer questions about local transmission versus imported cases, rates of epidemic growth, chains of transmission, genetic changes, and many, many more. But many of these questions require a tree as a starting point. And so we set out to put together a real-time phylogenetics pipeline, which could run on a daily basis, placing the UK samples into the global context. It's worth highlighting that before the pandemic, one of the largest phylogenetic analyses of an outbreak was the West African Ebola epidemic. And this included 1,610 Ebola virus genomes, which seemed like a huge number at the time. The method, broadly speaking, involved QCing the sequences, multiple sequence alignment using a tool like MAFT, masking problematic sites such as highly homoplasic sites, building a phylogenetic tree with BEAST, and then combining that tree with metadata to get to infer answers to questions. But by March 31st, 2020, the number of global sequences of SARS-CoV-2 had already exceeded this. So it was clear very early on that our phylogenetics pipeline would need to process sequences on a very different scale. And some of these parts of this overall sort of method would not scale well. In particular, multiple sequence alignment scales with the square of the number of sequences, and the largest BEAST trees had only included a few thousand sequences, and we knew that that would not scale to tens of thousands or even hundreds of thousands. On the plus side, one of the characteristics of this outbreak was the intensity of sequencing and the relatively low rate of evolution of the virus. So the result of that is that we see a lack of diversity between sequences. Even now, recombination is not yet a problem in terms of being able to handle some of these issues with pipeline development, and insertions are more likely to be sequencing artifacts than genuine variants. Plus, we have a reference that is pretty much the original sequence, or pretty close. And so instead of multiple sequence alignment, we can use pairwise alignment with Minimap2 to the reference and combine those pairwise alignments together with Go Faster, written by Ben Jackson. The problem of building trees, we've replaced with maximum likelihood trees. And I'm going to talk a bit more about how our tree building has changed throughout over the next few slides. Given the computational resources required, it made sense to perform the phylogenetic, the Datapipe and Philopipe sort of processing centrally, and to use it as a basis to answer the different questions. In practice, there is a lot that goes on under the hood in Datapipe and Philopipe, but roughly it boils down to this. We clean and QC the data, de-aligning sequences that some sites align before they submit, and checking for things like samples dating from 1980 or an admin 2 of city centre. We perform lineage typing with Pangolin and classify Vox and buoys with Scorpio. We deduplicate, removing duplicates that have duplicate sequences by COG-ID, and we flag samples that are longitudinal samples from the same patient. We align and filter by the proportion of ambiguous bases and trim the 5' and 3' ends from the alignment where sequences tend to be less reliable. We call all variants for each sample, and we combine all the UK data with those from GISAID, which have been processed very similarly. We then build a maximum likelihood tree and use this to infer UK lineages and phylotypes, which get added back into the metadata for further analysis. This pipeline's had to adapt considerably over time to scale. The first phylogenetic analysis of SARS-CoV-2 involved 11 genomes, and that was back in January. But by the time of the first SAGE report, 12 days after the start of COG-UK, there were nearly 1,000 global sequences. And this here shows a screenshot from that report of the microreact rendition of the tree. We quickly found that we had to split sequences and build subtrees for well-defined clades, which could be grafted back together. So instead of building just one tree, we built 3 trees for the A, B, and B.1 lineages and stuck them back together again within the pipeline. And then we had to replace IQ tree with fast tree. That happened fairly quickly as well. But this approach seemed to work well for us for a little while then, with new splits added when necessary. And then by January, we were again struggling to scale. We split the pipeline at this point into the two current pipelines so that people could get the lineage alignment and variant calls sooner, and started to restrict the number of sequences in the tree. And it's worth saying we also did a lot of refactoring at this point to remove inefficiencies like dependency on the pandas Python library, which was causing us big issues. Shortly after, we started using Usher to provide daily updates to an existing tree, and that really has transformed our ability to produce trees that are updated regularly at the scale that we're at now, with 4 million global sequences and 1.5, well, more than 1 million sequences from COG-UK. So the current tree, I think, has more than 1.5 million tips in it. So we now have a pipeline to generate a clean data set and tree, but we want actionable information from it. How do you get useful information out of more than 4 million sequences? We need a way of condensing all the information into something usable, interpretable, and reliable. So thinking first about local investigations, such as local outbreak analysis of transmission chain investigation, Anya and Verity created CIVIT, which is a tool to pull out the relevant bits of the tree and metadata and let you tease apart relationships between a set of sequences. It generates a printable summary report with a range of options to give you different kinds of information that might inform an outbreak. This is used already by many people. Researchers and clinicians across the UK, including at the Royal Infirmary in Edinburgh, are running it routinely to investigate local outbreaks, rule out cases of transmission, and to supplement and inform contact tracing efforts. And because they can run it themselves based off the central analysis, it means they can access that information fairly quickly, which is a huge improvement from sort of delayed ad hoc reports, which were made at the very start of the pandemic. Until recently, this worked by first identifying the local context of the query sequences in the tree, i.e. the most closely related sequences, and defining a catchment area. And more recently, within CIVIT 3, the catchment is drawn using a tool called Go Faster, which avoids the tree altogether and uses pseudo-tree-aware snip distances. But again, the expensive step is pre-computed centrally before people run the pipeline, before people run CIVIT. Seem to have stalled. Okay. Once catchments have been found for query sequences, it builds a local tree and then produces a report, which is highly customizable, showing things like we've got in the top left-hand side, we've got the available metadata for the sequences of interest, including private metadata that might be held by the hospital or site, but is not shared with the consortium, such as ward or additional patient information. We've got summary of catchments. We've got tree options. So here, the sequences submitted fell into 2 catchment areas. We've got 2 sets of trees and reports. On the left-hand side, you've got one, and you can see that they've decided to try and color by the ward status. And on the right-hand side, you can see, again, a similar one for a different set of wards. We've got snippet information, which shows how the sequences vary on a sequence level. And I've got a few more examples here. You can also have plots that show the spatial or geographical information about how the different sequences fall on a map. You can expand and collapse bits of the tree that look relevant. You can add in your own query mutation that you're interested in and see how that falls on the tree as well. And this final one again looks at how the information compares to the local area with the pie charts on the bottom plot. You can see how the lineages that are prevalent in that area that's being investigated is the same or different to the surrounding countries. And this has proved hugely successful for allowing individual sites to gain insights from their own data. What about actionable inferences at a national scale? In collaboration with others, our group has done more in-depth analyses into the introduction and spread of the first wave, the Alpha and Delta variants. And these analyses have included tens of thousands of sequences. And then in the case of the Delta variant, we're approaching 100,000 sequences. And for these sorts of analyses, we need to be able to use a tool such as BEAST for a time-measured phylogeny. But even with downsampling, the sequence sets that we have for these analyses are 1 or 2 orders of magnitude greater than, for example, the Ebola epidemic dataset. So JT and others have worked on several optimizations which would allow this analysis to scale. The first of these optimizations noticed that the most expensive part of the analysis is calculating the likelihood of an alignment on a tree. And so they've got rid of the alignment and are using as input a maximum likelihood tree. And the second optimization was to constrain the tree instead of trying to integrate all phylogenetic uncertainty. And the result is that instead of scaling as the number of genomes goes up like the blue line, we scale like the yellow line on the left-hand plot and like the red line on the right-hand plot. And looking just at the right-hand plot, you can see a couple of dashed lines at the top of the plot. And the very top one of those is the memory available on one of our machines that we run these analyses on. Its name is Arctic. It's a 50,000 pound machine. And to do something like the delta analysis, you would have needed 9 of those and months if it worked at all under the old way of running Beast. But with these optimizations, it's possible to do in a week, which is a pretty huge improvement. So I showed you the first newspaper clip. The wall has expanded since then. So this is how it's going, but I think that I want to finish off by thanking everyone in the lab. And all these tools and pipelines have been a massive team effort. And thank you again to COG-UK and the Arctic network.
Thank you very much, Rachel, and thanks to everyone who presented in our session.